Greetings from IIT Kanpur. This is Shantanu Bhattacharya from the Department of Mechanical Engineering, and I would be talking to you about this fascinating subject of introduction to biomems and microsystems. Biomems, as you probably some of you are aware already, are very small micro to nanometer size devices, typically for bio applications. The full form of biomems is also bio micro electromechanical systems. So let's actually look into a little bit of historical perspective of this particular area on micro systems, essentially, which started all the way in about 1959 or so. And essentially the first snapshot of why it is important to miniaturize things and how they can be used for various applications came by none other than Sir Richard Feynman, the very famous Nobel laureate, through one of his lectures called "There is Plenty of Room" at the bottom, which was presented also at the annual meeting of American Physical Society. So basically in this lecture, Sir Feynman actually illustrated the importance of going small by looking at several different perspectives like microelectronics, MEMS or mechanical systems, and essentially the idea was that through going small, he suggested very many improvements over the existing processes and some typical phenomena which would be totally different than the corresponding macroscopic intuition of people. So essentially after his lecture, there was a rapid technological development in the area of miniaturization of electronics to begin with, and this is also illustrated, as some of you may be aware, by the famous Moore's law, which states that the integration density of microelectronics essentially is almost at a rate of doubling every 18 months. However, it is limited by the resolution of the process called photolithography, which is essentially analogous to the conventional photography regime, and because there is a limitation that photolithographic processes cannot go below a certain level, let's say about 100 nanometers or so, this law may slowly change to doubling in every 24 months rather than 18 months. And so there are a lot of developments in this area after this thought-provoking lecture by Sir Richard Feynman, and essentially the whole silicon technology came up because of this lecture. Miniaturization of small components like transistors, small circuits, integrating them into a very hugely density oriented platform, it became kind of the technological advancement. Now silicon processors, because of this slowing down rate, going smaller and smaller, could only be limited, and so therefore some of the processes from silicon technology were really scrapped off, and this new area of micro devices or micro electromechanical systems really emerged because these processes which were left from the silicon industry were kind of accumulated together, and they were used for different applications, and as such this field developed as microsystems technology. As the field rapidly developed, there was an integration of fluidics, optical components, a lot of sensing mechanisms, etc., into this microsystems technology, and the whole domain shifted from really just the electronic sensing to all this fluidic or optical sensing, etc. So silicon processes also used in MEMS applications got to a certain maximum usage level, and in 1980s really for the first time probably the usage of these technologies, these microsystems technologies, were made for realizing microflow sensors, micro pumps, micro valves. Essentially all the microfluidics part came as late as 1980s, after the mechanical aspect of microsystems was thoroughly explored prior to that, starting from 1970s. The concept of integrated bio detection systems kind of merged with this microsystems technology as late as 1989, when this famous paper by Andreas Mans was presented at a plenary lecture, which was also known as the miniaturized total chemical analysis system. So, this essentially was presented in the International Conference of Sensors and Actuators, and this really changed the whole paradigm, because now people started realizing after this lecture what can be a very important potential of these microsystems research onto applications like sensing diagnostics, etc. And further kind of wood to the fire was promulgated by this famous project on human genome, which started in 1990 to 2003. This was probably one of the largest research initiatives done by United States Department of Energy, and the whole idea here was really to figure out or to identify about 20,000 to 25,000 different genes with about 3 billion chemical base pairs associated with these genes in the whole human body. There are about 360,000 different kind of cells within the human body, and you can think about it, how many base pairs, chemical base pairs of the DNA could really code these 360,000 different cells. So essentially it was a very large task executed by US and 7 of the other collaborating partners, including UK, Japan, China, France, and some other contributors. And the idea was to develop a whole gene pool where all this information would be packed together. And so Andreas Mann's paper in 1989, followed by this huge requirement of chemical analysis of molecular stuff like DNA, really made a very fast pace to the development of this whole area of bio-MEMS. So I would like to go to some of these scaled objects as to where what can be placed in microsystems technology as applied to diagnostics and detection. And if you really look at the different size ranges, it can be classified from 10 to the power minus 18, which is auto, all the way to about 10 to the power 9, which is giga. And there is a sequence of auto, femto, pico, nano, micro, milli, centi, deca, hecto, kilo, mega, giga, so on, so forth. So if you look at some of the volume scales in terms of the length scale, you can find out that if the length scale is in probably meters, the volume scales could be as high as about a liter or 1,000 liters. Okay, you go a little bit down to 1 millimeter and so of length scales, the volume being cube of length goes to as low as about a microliter or a milliliter. If you go further down to about 1 micrometer, which is about 10 to the power of minus 6 meters, then really the volume goes very, very less, about 1 picoliter or 1 femto or autoliter in that particular range. So if you look at some of the conventional fluidic devices, really it is in this area of about liters to about 1,000 liter volume scale. Typically the total amount of blood which is within us also ranges in several liters, and it is pretty much in this particular length scale. Human hair, on the other hand, if you consider the volume, it is close to about 1 nanoliters. The diameter of a human hair is typically about 100 microns, or about 10 to the power of minus 4 meters. A bacterial cell would have typically volumes varying from an autoliter to about a microliter or so. And if you go a little bit further down, these smoke particles, viruses, molecules, and these other so-called trace objects, they all fall in the sub-autoliter domain. Similarly, if you look at some of the micro devices which have emerged over the past so many years, for micro analysis systems, typically the length scale that is used is about 1 millimeter or so. Volume scales is about 1 microliters. Micro needles, which I would be illustrating again and again later in my lectures, is one of the major breakthroughs that probably this field had for painless drug delivery applications. They range in the range of about 1 picoliters to about a nanoliter. All nano devices, applications of nanotechnology, come again within about 1 nanometer to 1 micron range. We call it this sub-micron, and essentially it is again in the range of about autoliter or less. So micro pumps, valves, flow sensors, or micro filters, they really come in the range of millimeter to about microns. And so essentially in this area or in this subject, we would also be dealing with some of the devices which are more in this micro domain and can be used for various bio applications. So let me just introduce a little bit of the course syllabus for this overall course, and I would time and again pull up some slides to explain a little bit detail about the different aspects of this course. So, the first part of the course would include an introduction to biomems, biosensors, basic biology and microsystems technology. And so, essentially this would be very basic and definitional aspects of these different areas. We will try to define biomems, bio-nanotechnology, microfluidics, biochips and biosensors. And we will have a brief introduction to device fabrication, particularly using silicon and polymer. Biological moieties like bacterial cells or viruses, or in fact any other molecules, they prefer to have a happy environment when they are around carbonaceous materials. Polymers having carbon rich content form a very good alternative to such MEMS devices, and they can kind of sometimes replicate silicon devices with a much easier fabrication technology and can serve as excellent devices for handling these biological materials. We will also look at some of these aspects of sensors, transduction and performance factors. We will try to define sensor as a model and then look at some of the various aspects of human body like the eye or ears as a sensor, and we will try to model that according to the model of a sensor. And then we will have some introductory concepts into cell biology, DNA, protein, chemistry, etc., so that you can get a little perspective of what is it that we are really trying to detect through these biomems or biosensor kind of devices. So, if you look at the definition of biosensors, it actually comes all the way back from about 2000 by this work from Woodin and Cullum, which talks about biosensors as analytical devices that combine a biologically sensitive element with a physical or chemical transducer to selectively and quantitatively detect the presence of specific compounds in a given external environment. So, the role of a sensor is really to pick up a particular analyte of interest over the many analytes which are there in the environment, in an external environment, using some kind of a recognition mechanism which can transduce a chemical or a physical signal into a certain readable signal which is quantitatively able to be gauged and which can give an idea of things like concentration, the intensity of the particular material or signal that we are investigating through the sensor, etc. On the other hand, biosensors need biochips to be detected, and this whole domain is really biomems. So, biochips can be defined as microelectronically inspired devices that are used for delivery, processing and analysis of or detection of biological molecules and species, as indicated by Rashid Bashir in the year 2004. These devices are used to detect cells, microorganisms, viruses, proteins, DNA and related nucleic acids and small molecules of biochemical interest and importance. So, essentially it is a promulgation of all this microelectronically inspired devices and device technology on a single platform for delivering a particular analyte of interest, processing the analyte of interest and analysis and detection of the signals that occurs, and the analyte of interest in this case is mostly biomolecules. So, the next part of the course would cover some introductory concepts on microfluidics, where essentially we would be studying fluid flows at the microscopic length scale. And here we would start with studying fluid as a continuum, some fundamental concepts in that region, some differential analysis of fluid motion, mass and momentum conservation. Essentially we will be dealing with the Navier-Stokes, all the three equations, actually two, equation of continuity, conservation of mass, conservation of momentum. The energy equation is not very critical in case of the micro domain, because we hardly have much of heating effects, and microfluidics so far developed has not really looked into much of heating related or heat transfer related issues in microchannels. So, therefore, we will be confining ourselves to only the first two equations. So, we will do continuum mechanics at a small scale. We will study gas flows, liquid flows, boundary conditions, typically low Reynolds number values flows. Microfluidics is essentially about all this domain. The Reynolds number is often 100, very often less than 0.1 in such devices. And then we will be talking about entrance effects, which is a critical, very critical effect if you consider microfluidics. We will be talking about surface tension, which is essentially surface related activity, which gains much more prominence over the volume effects. And then we will be talking about things like electrokinetic techniques, which come more prominently on the micro domain as opposed to the macroscopic length scale, and some associated techniques like electrophoresis, electroosmosis, dielectrophoresis, etc. So we will also try to apply whatever knowledge we gain in microfluidics by studying flow control devices like micro pumps, micro valves, and we also do device building and characterization, and also study micro mixers in some details. So essentially, as probably as mechanical engineers, we are all aware about the dimensional form of the Navier-Stokes equation. The whole idea is that at the micro scale, how you scale these equations in a manner that especially the conservation of momentum equation becomes time independent in nature because of the low Reynolds number value here. So as you see here, the right hand side really of this particular equation is independent of the time dimension, and if you look at the left hand side, really you have the Reynolds number coming out of this whole mathematical expression and the time factor which is within the brackets here. And so assuming that the Reynolds number in microfluidics is of the range of about 100 or less than 0.1 at times, we can consider this whole on the left side of the equation to be just give me a minute. So the whole left side of this equation becomes time independent. Reynolds number being close to 0, you can neglect the LHS of this equation. Essentially what it means is that the equation typically becomes more time independent. This is of great importance, especially in the area of micro mixers, where we have situations where there are two flows coming within the mixer and hardly mixing. So if you have exactly opposite time behavior, that means from the output to the input side, you should be able to typically extract the flows because they are time independent. They don't change with time. So some of these concepts we'll be discussing later as we go in more details along the course. So this slide is something that I keep on showing about microfluidics. Here this simulation here is really picked up from the Whiteside's group, which talks about these 5 different dyes or 7 different dyes which are moving along the small channel. And as you see here, that if they move, even if they move along a certain amount of length, they are able to be extracted as non-mixed components, and there is no change in coloration really. And this is because of the micro scale effect. We will be actually studying these in a lot detail later on. The third part of the course here is designed to give you an insight about some of the fabrication processes that we use for such microsystems technology. Essentially, microsystems really started from processes generated by the silicon industries. So the base material there, which was mostly used, was silicon, and essentially some of the processes for silicon include things like optical lithography, photoresists, vacuum science, and plasmas form a very important component of fabricating, because plasma systems are off and on used for things like etching on silicon, where you can create micro features and structures, and wet etching techniques where you use acids typically or sometimes basic compounds for etching selectively, and combining this with optical lithography enables you to etch at a scale which is very, very small and defined and can be defined by light. You also have processes related to evaporation and sputtering, wherein metal target is heated to its boiling point so that it starts evaporating, and the metal vapors are captured on the top of a silicon surface, where they get a thin coating which is a few hundred nanometers in thickness, and that can be used for different applications. We'll also talk a little bit about polymer fabrication processes, and as I illustrated before, the subject being a little bit biology oriented, and essentially about detecting and diagnosing different biological moieties, they do prefer to be kind of behaving well when they are in polymeric structures. So, we will be talking about some basic fabrication processes in polymer systems, things like replication and molding, also developed for the first time by Whiteside's group. We will talk a little bit about microstereolithography, where we will talk on a three-dimensional basis how we can formulate these microstructures or shapes. We will talk about a little bit of biocompatibility aspects of these polymer materials, something which makes them really have a cutting edge over the other inorganic kind of materials like silicon. And then we will also discuss various soft lithography techniques like capillary molding or nano imprint lithography, and so on and so forth. So, typically if you look at all these different fabrication processes, they have been categorized into two broad kinds, the bulk and the surface micromachining. And essentially bulk micromachining is all about subtractive etching, where you are pulling off material from within the bulk or the volume of the material, and surface micromachining is about building features or structures on the surface of something. And these are the two classifications into which the whole fabrication technology can be broadly divided. And then we will have a little bit of introduction to MEMS design, of course. Let me give you a little bit of flair of what optical lithography really means to some of you who are probably new in this area. So, lithography is essentially something which is analogous to photography. So, in a photography process, what happens? There is a camera film, and this film has been exposed, and you have a feature or something which is already there, and then you use this as a mask for preventing light from falling onto the surface of a photo paper. So, you have, let us say, a light source somewhere here, and you have this film material which is essentially having some vias and through passes for the light to go in these particular regions and fall on a photo paper. The photo paper here is a material which gets actuated and changes its chemical property on exposure to light. So, when such a thing happens, then whatever is there on the film essentially gets replicated very finely onto the photo paper, because the photo paper, wherever it exposes or get exposed to the light, changes its chemical property, and it can be visually distinguished from its other surrounding areas which have not been exposed to light. Lithography essentially is a similar kind of process. So, you start with something called a material where the base material or substrate where the processes are to be incorporated. Most of the time it is silicon to begin with, and then you essentially spin coat an adhesion promoter layer which is used for promoting the adhesion of a photoresist. What is a photoresist really? Photoresist is something, it is a chemical which is essentially liquid in nature, but then you have a photo definability in that chemical. So, if you have a light source falling onto this chemical through a certain masking material, it should change properties, and there should be features defined just like the photo paper as I talked about before on the top of this particular film. Okay, so, this resist material is coated on the top of the adhesion promoter, as you can see in the process 3 here. So, the adhesion promoter is essentially a cross linker between the surface and the resist and prevents the film from getting broken or the film from getting lifted off. Okay, and so it essentially serves as a binding material for the whole continuous photoresist film and to be there on the silicon surface. And the next process of lithography includes the masking, and essentially this masking is nothing but the analogous of the film in the camera process, the photographic development process. A mask is a black and white material where or rather a transparent and a black material where the features that we want to define onto the resist are very well laid on the top of the mask. Okay. So, thus giving openings or vias for the light to go in those regions where there are features which are printed. So, if you look at this figure here, this black portion really is the mask, and you can think this as that there is a source of light which is on the other side here in this region, which is emanating these parallel rays, and of course there is a lot of optics which goes from this particular light source all the way up to the film. And then when the light falls into this film, the areas which are transparent, typically these would give away to the light, so that the light falls only in these defined zones where the light can go through the mask onto the resist. So there are two kind of different resists which are available. One is classified as a positive tone resist, another is a negative tone resist. Essentially a positive tone resist is something wherein wherever light falls there is a tendency of the resist to move away. Okay. So essentially you are having a debonding action here. So you can think about it as that, you know, this part of the film which the light was not exposed to has is in a cross bonded state, and the portion like this and this where the light came in from the mask earlier is kind of moved away and it gets debonded. So you have these features imprinted onto the resist surface as vias and trenches. The negative resist on the other hand is the other way round. So generally it is debonded in these regions and then it is cross bonded wherever there is light falling on the top of it. And so therefore in case of a negative tone resist there is a tendency of the resist to stick on to the surface as light falls from the mask onto the resist surface, and you develop away of course the unbonded portion, and so essentially you are left with these features onto the resist surface. So we will be doing a lot more of this as we go into active MEMS fabrication. How do you do photolithography? So essentially you start with a substrate, you start cleaning the substrate, make it a super clean, super finish surface so that it can adhere to the resist film very well, and resist being a liquid material, you spin coat the resist onto the top of the wafer using just a normal spinner, rotate it at a certain RPM, pour a certain amount of resist so that it can go and quickly cover the whole film in a planar manner, and essentially then you do what you call the pre-bake or just heating the photoresist. So if you look at the photoresist, most of the resists do have a carrier solvent, which is an organic volatile material, and essentially the solvent has to be evaporated for the resist to be deposited on the top of the plate. So essentially as the soft bake process is executed, the resist gains its strength and its ability to keep on the surface, after which you actually expose using a mask, and the mask can again be made by some other mechanisms like this electron gun, scribing through a film, maybe, you know, a metal film which is well laid out on a surface, or using a black and white transparency mask even, where you can print at a very high resolution and can be able to produce features about 50 microns or more using such a mechanism. The files which are used for printing these masks or realizing these masks are generally CAD defined, so you have to have a design package associated with for laying out the mask or, you know, designing the mask. So once the mask is prepared, you expose the resist, and then you have the step of post exposure bake, which is because, you know, the exposed resist needs to be fully initiated. So there is a tendency of heat to sometimes catalyze the bond formulation or the bond breaking process. I just showed you in the last slide how a negative and a positive tone resist behaves. So in a negative tone resist there is a cross bonding as light falls on the top of a surface, and a positive resist it is vice versa. So this cross bonding action is sometimes catalyzed by heat. So there is a tendency of heating the exposed wafer to another step here so that the resist can get fully catalyzed. If you can give sufficient time instead of heating, the same effect can be felt just with time, but heating is normally done to quicken the processing of the resist. You then develop the particular resist. So in case of negative tone, the areas which are unexposed go away, and in case of positive tone, the areas which are exposed to the light and debonded in the process go away. And so in either case there is this formulation of the whole the design on the mask on the top, or the transfer of the design of the mask on the top of the resist. You sometimes do a post bake for doing a better job with the material coming out, and here in order to ascertain sometimes whether the resist is properly developed, you use some reagents, as I will be illustrating later, so which leaves a mark if there is any unbonded resist which is left over on the surface, and so that gives you a feeling of whether the whole area which has been exposed is the only area which is remaining or which has gone away, depending on the type of resist. You then can do, of course, a lot of different things, like if you want to use this resist as a mask, as a sacrificial mask, you can use this to etch off the material by creating a layer and opening these vias at different places through which you can give this etchant and can etch relatively thin films of silicon dioxide, nitride, some other materials, etc. And then of course you can strip the sacrificial photoresist, because dissolving in in an organic solvent like acetone would essentially eat away the resist which is left over on the surface. So this entirely is a process of photolithography, and we will be looking into a lot more details and other aspects of this process as we go along in the microfabrication. The fourth part of the course is really the most interesting part, based on some practical applications of these technologies like lab on chip, microsystems technology, and biomedical chemical sensors, etc. We will be discussing specific cases wherein we can do integrated gene analysis using such systems. We can use these systems to detect cell growth as if they were growing in a petri dish in a laboratory, and we can perform all kind of things like lysing, analysis of pathogenic bacteria, culture growth, etc. Integrated trapping of these to increase the concentration locally sometimes into the chips, and we will also do a little bit of electrochemical and optical labeled and unlabeled detection of such moieties, and there would be mostly discussion on review articles and papers in this particular module of the course. So I would like to just give a small illustration here of what can a typical lab on chip device do. So in this particular module, for example, back from one of my earlier works, we have been developing a lab on chip module which does an integrated approach to detection and diagnostics of bacterial cells. So you have a fluidic port here, as you see, wherein you actually flow the analyte of interest, which contains let us say food pathogenic bacteria, pathogenic bacteria of some form. So the first step on this particular chip is really the localization or concentration of this particular analyte of interest onto a very small volume. So we use a method for this called dielectrophoresis, where alternating electric fields are used to rapidly trap and capture, and we will be doing this process a lot in details later on, the flowing cells through particular microchannel and pre-concentrate it in a very small region. We then use such such moieties or cells by coating a layer of antibodies on the top of these electrodes, which would actually selectively bond to some of these bacterial pathogens, and that way there could be an identification or selection on the various pathogens that are flowing in. So typically the unbonded ones would be washed off later, and the ones which remain are the ones which also adhere to the antibodies coated on the top of these electrodes. We simultaneously also do impedance spectroscopy, where in a micro scale, as we flow a growth media in these captured cells, the cells grow by themselves and it changes the pH of the medium, and so there is a change in the impedance parameter in this particular zone or region, and that gives also an aspect of viability of the cells. So viability is also the livelihood, the ability of the cells to be the ability of the cells to be live versus dead. Okay, so whether they are living or not can be found out by a change in just impedance signal and electronic signal. In the following module, we do a temperature mediated lysing of these grown cells, and we extract the genomic information here, which is trapped within the bacterial cell. I hope most of us are aware probably that the bacterial cell does not have a nucleus on its own. It is just a cell wall which encloses all the cytoplasmic content inside that wall, and all the genetic material is sparsed around that cytoplasm. So essentially a rupture of the cell walls would expose all the genomic information or all the molecules related to the genetic information of the material externally, and we run what we call a PCR, which is essentially polymerase chain reaction. It's a very famous technology which was developed in the year 1982 by Mullis et al., wherein a segment of DNA is taken and chemically it is processed through a set of steps so that you can amplify the information on the molecule. And by information on the molecule, what I mean is essentially if you look at the DNA from the Watson-Crick's model, it is a set of base pairs, and there is a set of information of those base pairs on the molecule. And so therefore through the PCR process, the particular information or the particular sequence is replicated or amplified many times. Millions of copies of the DNA are generated, and then if you could somehow transduce the information by converting this chemical information into an electrical or an optical signal, then here we have an identification, a molecular identification basis of the particular pathogen of interest. So all these processes that I have been talking about are integrated onto a single chip. This is of course the output side for the material to go out, and this particular chip can very selectively, specifically and sensitively detect bacterial pathogens on different samples of interest. So essentially this is something like what a total integrated system can do, a miniaturized total chemical analysis system can do, and this is also known as lab on chip kind of technology. Welcome back. I would like to now illustrate from where we left last day, and essentially I would first like to kind of review my previous lecture on the introduction topics, which have been also the first two lectures of this session. So in the first module we were talking about introduction to biomems, bio-nanotechnology, biosensors, biochips and microfluidics. Some of the definition aspects of these were covered. If you remember, we also talked about sizes and scales, wherein we demonstrated the different sizes of some of these biological moieties or entities on one side with scale or dimensions, and we also compared some of the MEMS or microsystems technology related devices which are available commercially and otherwise, and tried to prove or give an illustration or a reason why it is needed to merge these two worlds of microsystems in biology. We discussed a little bit of chemical and biological sensors, some definitional aspects in our last lecture, and then we tried to understand sensor as a model where there would be a recognition element, there would be a transduction type, there would be essentially a processing of signal of one form to another, and it would read on an analyte, give a signal of some other kind which is readable, and would be fed into a signal processor, and essentially the signal processor would be able to tell or distinguish what concentrations or what types of analytes are there in the particular sample of interest. So the whole organization of this information was studied in perspective of different sensors which the human bodies have. Human body is essentially our nose, ears, eyes. These are all different forms of sensors, and so we try to illustrate these two, nose and eyes, as sensor models. And what we found out also was that if you talk about nose, we have the analyte of interest, which is probably the particulates or the air around us, which we are trying to detect. And then we had a mucous membrane, and there were some receptor cells, and there were this fine proteinaceous materials called cilia on the surface of the cell, which would cause electrochemical reactions from the various gases that you inhale. And due to that electrochemical reactions, there would be a series of electrons which are generated, which are again transported by the back end nerves all the way to our brain. And brain was a signal processor essentially. So we talked about the mucous membrane as the transducer, our nerve cells, the mucous membrane as the recognition element, our nerve cells as the transducers, and essentially the brain as a signal processor in that case. Similarly, in case of eyes, we described how sensation is felt by conversion of a cis retinal compound into a trans retinal compound, and how a cis retinal is actually also isomerically conjugated to an opsin protein called rhodopsin present on the retina of the eye. And so we basically talked about this retinal as the recognition element in that case. Again, the nerve cells which would convert the optical signals or the light signals into an electrical response, and the brain as a signal processor. So we also considered the various aspects of sensor design, wherein we discussed the various things like recognition elements, where this could be biological or it could be chemical recognition elements. We talked about transducers as substances or materials which can change the form of signal from one to another, from chemical to optical, chemical to electrical, etc. We also talked about what immobilization could do and how important it is for recognition elements, and this area would be taken up further today. Actually, this is where I would like to start on today from, and then we also talked about some performance factors. So let us look into a little bit of details about what can transducers do. So essentially, definitionally again, transducers are detector devices. Okay, so they detect a particular analyte of interest and convert them into a readable signal. So there are several examples of transducers. If you look into analytical methods in chemistry, photometric means are probably one of the best used transduction mechanisms, wherein the change of signal is really through a beam of light, and it formulates spectroscopic or colorimetric scheme. So when light of a certain wavelength goes through a material, there is either an absorption at a selected wavelength where there is a change in intensity, and the output light essentially contains less intensity of that particular wavelength. And so this absorption can be owing to various ways and means. One of the reasons why absorption happens is bond vibration. Okay, especially if you are talking about infrared based spectrometry, there is a tendency of whenever beyond red, whenever you hit a particular wavelength of light onto a material of interest, there is a vibration between the molecular bonds really, and a part of the energy is dissipated, and it's also a function of the bond energy. Okay, so various bonds, like let's say silicon dioxide, nitrogen oxide, and different kind of bonds or different kind of chemistries can be identified very well by that means. So essentially that is nothing but a transduction process. So you have an ambient energy, and it converts into a mechanical energy of some sort, and the ambient energy gets changed, and that contains the information of what that element is that you want to sense. Okay, so that is also another kind of photometric transducer. Colorimetric methods, again, we saw in the last lecture how an ELISA mechanism works. So you have a chromogen of interest which comes in, you have an immobilized enzyme through a secondary antibody onto a primary antibody, which essentially we are trying to detect in a patient's serum, and these are immobilized by immobilizing certain antigens onto the top of a plate where you have this whole mechanism. And so the moment the chromogen comes in contact with the enzyme, there is a change in the absorption wavelength, so there is a change in color. So they are known as colorimetric methods. Some of these can be also used as sensors, because there are certain thermochromic dyes where very accurately, up to an extent of almost about a millionth, about a thousandth of a degree, you could detect a small amount of temperature change by a corresponding change in the absorption spectrum of a dye. Okay, so essentially that's also some kind of a transduction effect. You are changing temperature there in terms of a color. Okay, most of the transduction, however, in this kind of applications is electrochemical in nature, and one of the reasons why electrochemical is preferred over the various other methods is because if you look at the construction of devices which can do electrochemical sensing, they are really very simple and inexpensive to make. It's all about putting a reference electrode and a measurement electrode, and there can be either a potentiometric or an amperometric kind of measurement. I'll just illustrate all these different mechanisms briefly in the next slide. We also are kind of having an advantage in terms of the low cost of manufacture of such methods of transduction. And what the most important factor could be probably is that it is very easy to integrate with microprocessors, because essentially every conversion in electrochemistry is about a chemical into some kind of an electrical signal. So it is very well compatible with microprocessor technology and microelectronics. So it makes the readouts of such systems very manageable, and so that's one of the reasons why transduction in most of the times is a preferred mode of transduction is electrochemical. So there is a new area which is developing in the recent times, and these are these photon driven devices, and they are emerging. So there is a gradual shift towards optical fibers from electrochemical sensing mechanisms. One of the reasons why that is so is that photons essentially are much faster than electrons are, and sometimes you can maneuver and manipulate these more easily inside devices. So there is a general impetus. In fact, even slowly some of the electronic devices are converting into photonic devices over the recent times, and so therefore there is a impetus in the photon driven devices. Another emerging area for sensing is micro mass controlled devices, which is based on piezo kind of mechanisms. So there is a vibration frequency of a certain crystal, and there is a voltage generated, there is a voltage given, and there is a corresponding vibration. And so if there is an absorption of a certain analyte of interest on the top of such a vibrating platform, there is a natural tendency for its resonant frequency or natural frequency to shift, and you can back calculate the amount of mass which is absorbed onto the surface by just calculating what is the shift in the frequency. So these kind of transducers are also coming up of late, wherein piezo crystals are rapidly used for detection of femtogram size masses. So if you look at electrochemical transducers, really there are several of such transducers. One common scheme is potentiometric transduction, which involves the measurement of EMF of a cell at zero current. Okay, so you have a situation where there is hardly any current flow. You have a monitoring electrode which just monitors the potential of a particular solution with respect to a reference electrode, and as the concentration of the analyte of interest is changed, the EMF in this case varies proportionally to the log of the concentration of the analyte. So these techniques are known as potentiometric techniques. Other set of electrochemical transducers are voltammetric in nature, where we talk about a redox system. So from our basic knowledge of chemistry, you probably already know that redox system is kind of a coupling mechanism where there is a reduction of a species and a simultaneous oxidation of another species at the cost of the reduction. So and it's essentially an equilibrium process, and there is a shift of equilibrium if you change various parameters on both sides of the reaction. So this can be a mechanism to be studied, or this this whole idea of where a particular material is reducing or oxidizing on the voltage scale can be a very good mechanism of studying the type of material present in the analyte system. So when you these kind of voltammetric kind of transduction modules involve the change in potential of such a system. A voltage is applied, and there is a rapid scan of an increasing voltage that you apply to such a system. So at a particular voltage there is always a reduction of the reduced species. So what would happen if you look at the current response? So essentially, let's say you are trying to scan the voltage, give me a minute, across, this is the current I and this is the voltage, and you are trying to rapidly scan the voltage from say 1 volt all the way to about 6 volts maybe, and there is a reduction, there is a species which gets reduced at a particular voltage, let's say about 4 volts. So essentially what would happen is that the current, as you monitor the current, it grows ohmically, and as soon as it hits the reduction area, there is a sudden increase in the current, and that's because there is an electron release here. Okay, let's say this is an oxidation. Okay, this is an oxidation process. So there is a sudden electron release. Zinc, for instance, becomes Zn plus 2 and 2 electrons. So the overall current value, which was ohmically varying up till this place, would suddenly increase because of that. Okay, and the particular voltage at which this current increase happens gives an indication of what species is present within the analyte of interest. Similarly, you could have a backward voltage more towards the negative side and get a scan here where there can be reduction at a particular point of time. So, this whole principle of scanning the voltage or rastering the voltage on certain ranges and trying to study what point a species oxidizes or reduces as so as to detect the particular species of interest is also known as cyclic voltammetry. It is a very common technique in all electrochemical sensing operations, and here in the voltammetric techniques, the peak current that you get, this peak value of I here, is proportional to the concentration of analyte of interest. So, if you have an oxidant which has a certain concentration and a changing concentration, let us say it is an increasing concentration. So, there would be a corresponding peak change, as you can see here, of the current on different concentrations, on increasing concentrations. So, that is what a voltammetric system would do. Another very interesting form of electrochemical, and this can be more an electrical than electrochemical transduction process, is known as conductometric or impedometric sensors. So, essentially what this kind of sensor does is that, because owing to the absence or presence of an analyte of interest, there is a sudden change in the conductance of the medium or the impedance of a medium. Okay. So, I just would like to take you back to your class 12 days when you were studying impedance. So, essentially, as we all know, impedance is the analog of conductance when we talk about complex, it has a real part and a complex part. The real part is a resistance, complex part is a frequency dependent resistance to the flow of currents or the path of currents. So, impedance essentially is when we talk about an alternating or a variable voltage source, as opposed to probably conductance, which is more related to a DC direct current source, or a static voltage which is not changing with time. So, impedance is a better word here because when we talk about electrochemistry, we are essentially meaning an electrode which is dipped within a particular solution, and once a metal electrode is dipped within a solution, there is a formulation of something called a dual layer of charges. I will be illustrating that in a little bit in more details as to what this dual layer is or how it comes from. But the idea is that because it is a charge separation, there is a capacitive component between the solution and the electrode, and it is best identified by a variable voltage, sinusoidally variable or cosinusoidally variable voltage, or in other words an alternating voltage rather than a direct measurement. So, impedance is a better term sometimes in comparison to conductance when we talk about electrochemical measurement. So, if there is a change in analyte of interest, there is simultaneously a change in impedance of the medium, and you can back calculate what is the concentration of the analyte, whether it has increased or decreased based on this impedance change of the medium. So, that is another kind of transducers. The optical transducers probably we have discussed in details. It can involve different optical phenomena like absorption, fluorescence, luminescence, internal reflection, surface plasmon spectroscopy, and light scattering. Let me illustrate a little bit of details about what fluorescence means. So, as we probably are aware that some of the objects or some of the chemicals have this tendency of showing a glow, okay, when exposed to a certain excitation frequency of light. So, a very commonplace thing to be seen in toys sometimes which glow in the dark is what you call as fluorescence. So, fluorescence happens because there is a change in the electronic states from the ground to the excited state and back with a certain loss known as phononic loss. Okay. So, essentially the whole aspect is that there is an increased yield of the light generated by and of course the light generated is of a higher wavelength and a lower frequency in such situations. So, there are certain dyes or chemicals of interest which show if a fluorescing behavior when an orbital state is formulated. Okay. So, if they come close to one of these biological moieties, let us say you have a DNA molecule or a protein molecule, and there is a fluorophore which comes very close to such a moiety, So, there is a conversion in its molecular states. Okay. So, the energy level, orientation, etc., changes, and then that necessitates this transfer of electrons. It generates a situation where the substance may be having a sudden increase in the yield of the light that it could generate. So, it glows, or it starts glowing, and that is a very important detection technique, an optical detection technique. Similarly, luminescence is another very interesting area where let us say firefly is essentially luminous. So, it becomes suddenly a self luminous situation, and there is a physical reason for that. We will be understanding in details a little bit later. Internal reflection again is another means of optical transduction. Surface plasmon resonance or surface plasmon spectroscopy is a very important mechanism which can do wonders by picking up very trace analytes. So, essentially what happens is that in these kind of devices you have a light beam which comes onto a get internally reflected because of a contrast in the refractive index of a surface with respect to the medium in which the surface is immersed, and because of this reflection, there is a certain depth over which the evanescent field would go from the surface into the medium. Let us say you have, let me illustrate it a little bit clearly, that you have the surface of a material, and there is a medium on the top here, and there is a beam of light which falls onto the surface, and the surface has a refractive index contrast, let us say N1 and N2, in a manner that this beam is totally internally reflected back here. Okay. So, if you look at the electric field, and as you all know, light is an electromagnetic radiation, there is a percolation, a steady percolation of this field into this medium here, and so there is a steady percolation of this field into this medium here, and this is also known as the lost field or the evanescent field. So, what it really depends on as to how much loss this particular incident light would face is based on what is this contrast of refractive index. So, let us suppose the surface absorbs certain antibodies here, okay, and on the top of it, it absorbs certain cells here. So, there is definitely a localized change in the refractive index close to the surface, and because of such a change, there is always a tendency of this the reflected beam of the surface to be varying in energy. Okay. So, there can be either a wavelength shift, or there can also be a change in angle, and so this essentially is what a surface plasmon spectroscopic system does. So, by this change in angle or the change in this incident in this reflective waves energy, you could figure out, is there an absorption on the surface, what is the kind of contrast on the refractive index that has resulted. So, if a certain concentration of cells is put in this particular situation, let us say you will have a different wavelength shift as opposed to a certain other concentration. So, this is a very unique transduction phenomena that can be used in most of these sensors or devices. Light scattering is also a very important aspect. As we all know, a medium can be turbid, medium like milk, let us suppose, that when you pass light through it from one end, it scatters the light in all different directions. That is why it probably looks turbid, it looks kind of milky, as we say. So, scattering again is in a sense a very interesting optical phenomena which can be used for doing a lot of sensing, particularly related to detection of cancer within skin, or even on the surface. Sometimes this principle of light scattering is very often used. Piezoelectric devices, we have probably talked about the change in the vibrating frequency or the natural frequency of vibration of a crystal as some material is adsorbed onto the surface. And the other means of transduction, signal transduction, is thermal means, and all these chemical or biological processes involve absorption or production of heat, and that somehow, if we can detect, can be a means of detecting the substance that is to be analyzed. So, as far as the transducers types go, these are pretty much what they would look like. So, I was talking about in the electrochemical sensing the double layer. So, I would just like to explain a little bit of how this double layer gets developed. So, let us suppose we have this piece of metal, let us say zinc electrode, which we are inserting inside a water or a solution which contains ions such as zinc ions. Okay. So, there are positive zinc ions in this solution, as you can see here, and you have this zinc electrode that you have just dipped within such a solution, and there is almost immediately a tendency of some of these zinc atoms on the top of this particular electrode here in this region to migrate rapidly as zinc ions into the solution. Okay. So, this happens because of the thermodynamics. There is always almost the tendency of local electron neutrality to be maintained. So, there is this famous principle of electron neutrality, which means essentially that if there is a charge buildup of a certain type in a certain region of space, automatically the opposite charge kind of comes in, and the particular region in space that we are considering tends to be electrically neutral in nature. So, if there is a charge buildup here, let us say we have a zinc sulfate solution and you have SO4 minus 2 and Zn plus 2. So, there is a charge buildup here near the surface of the zinc, and there is almost immediately a tendency of atoms to get ionized into zinc plus 2 and getting into the solution. Okay. So, this essentially is the reaction what happens. The zinc from the metal converts into zinc ions and goes into the solution and leaves 2 electrons. Now, what essentially happens because of this is a very interesting thing, that there is a certain density of negative charges which comes on the surface or close to the surface of this particular electrode, okay, as opposed to the solution, which is probably having these positive charges which have come out, okay, in this process. So, as there is a separation between the positive and negative charges here in these two regions, there is almost always a tendency of an electric field to get set up. The question is, why should the positive charges which have been liberated from the electrode onto the solution not go back and get neutralized? Okay. So, this can be explained by the fact that there is water in this particular solution, which is a dielectric material. Okay. So, if this dielectric comes in between, there is a separation, a perfect separation of the charge. So, there is a zinc atom which is separated by a small layer of this dielectric in between, and then there is this negative charge developing on this particular electrode. The question is, can this process keep on going? The answer is no, because essentially there is this process is diffusion driven, and after a while there would be a certain density of negative charge, and you are not pulling out that charge from this particular electrode, and this negative charge density would not allow any further electrons to be automatically inducted into the electrode, and therefore the process of zinc getting converted into Zn plus 2 automatically stops, okay, after a while. And what develops is this dual layer of charge, which you also know as the electrical double layer. Okay. There is a lot of interesting things which happen in this double layer, and essentially all electrochemistry is about the charge transfer process across this particular layer. So, the degree of charge unbalance produces an electric potential between the two phases, that is the solid and liquid in this case, and definitionally electrochemistry is really all about the number of charges that cross through this interface between the two phases, and we would be all interested in studying this charge transfer aspect. So, when you are talking about detecting an analyte of interest, let us say, which is this liquid solution, you have to be careful about how to maneuver this double layer and how you can actually keep it as an essential part of the system and still be able to get an accurate measurement of the analyte of interest, concentration, etc. So, the transduction part of it is more or less kind of discussed now. I would like to go to another very important part of this whole process of sensor design, which is the immobilization method, and we had already discussed before about that these recognition elements that we have been talking about, how these elements have to be mobilized on the surface of the sensor. Okay. So, the word immobilization really means the tendency of the recognition element to kind of conjugate or associate with the surface which is used for sensing the particular analyte. So, it is needed between the recognition element, let me put it this way, between the recognition element and the transducer surface. There are several ways and means of doing it. Physical absorption of the the recognition element is one aspect. Let us say we are talking about bovine serum albumin or BSA, and we want to there is a way of doing or putting a biotin moiety on the top of this BSA molecule. So, you have a surface here and you have this biotinylated BSA. Let us say this is the BSA part and this is the biotin part, which you have either commercially obtained or you have developed, and then you have an antibody that you want to immobilize on the top of this with a biotin end group on the antibody. So, there are protocols which can be used in biology to do this biotinylation of different materials. Okay. So, there is an easy way of putting a biotin moiety into a molecule of interest. Now, if you have such a case, you have a biotinylated antibody and a biotinylated BSA, and you can put something like a streptavidin moiety in between. This is one of the most famous streptavidin biotin molecular locks which are used in almost all biochemistry, and this can be a method of immobilization. But here the BSA can be made to physisorb onto the surface of interest. Okay. So, if you have let us say a silicon dioxide surface and you want to somehow immobilize this BSA, all you just need to do is to change the pH of the medium in a manner that there is a tendency of the SiO to kind of develop a negative charge and the BSA to kind of get protonated on one of the amine groups and getting an NH3 plus, and there is a kind of electrostatic attraction between the two, and this can result in adsorption of this recognition moiety, which is an antibody. We have been talking this before when we talked about recognition elements, onto this surface. Okay. So, physisorption is definitely one of the easy or most easy ways of immobilization of a moiety onto a surface. Another very interesting way is microencapsulation, where you take a particular moiety of interest and then try to kind of trap them between two membranes on the surface of the detector. So, you make a membrane which is electrically permeable. So, there is a, let us say you are developing an electrochemical sensor. So, you have to have a permeability of this membrane to the flow of electrons, right, because whatever reduction and oxidation processes are taking place within the analyte has to be read in terms of an electron flow which goes through the transduction module of the sensor. So, whatever recognition element is there is placed between two membranes. The membrane gives the mechanical support, it kind of lets the recognition element stick to the surface of the sensor, and at the same time is permeable to the flow of electrons. So, such aspects are very often useful for designing sensors, and we will be studying some of these aspects. Entrapment is another method of immobilization. So, the difference between microencapsulation and entrapment is that this microencapsulation is trapping between two membranes, and encapsulation is essentially a volume based trapping by using something like a gel material. So, I think let me just go ahead and give a little bit of basic on what a gel material is. A gel is something which is semi solid kind of material. You have enough liquid content in the material, and then you have a porous structure essentially which contains the liquid, and the porous structure gives the kind of mechanical solid support. For those guys who have been in some of the biological modules or biological laboratories they have done, you are probably well aware of the famous agarose gel, which is essentially something like gelatin, okay, like a jelly kind of a material. So, you have enough of liquid and also a solid phase mixed together in a certain ratio. So, you can entrap these recognition elements on a gel in a volume basis and apply that paste or polymer onto the electrode with the particular agent for doing the recognition trapped within this gel. There could be another way of immobilizing, which is just a simple covalent attachment, if such chemistry exists, of course, between the the particular moiety and the surface of interest. So this again is a perchance, or a it's a matter of chance that such a covalent mechanism or covalent chemical bond formation can be developed between the moiety of interest and the surface in question. Another very interesting mechanism where a lot of work has been done is cross-linking. Okay, so the difference between covalent attachment and cross-linking is that in this particular mechanism, the molecule, which is the recognition element, is directly covalently bonded onto the surface, and in cross-linking there is a ligand material, and ligand, by ligand we mean a linker molecule, which is essentially used, it's a bifunctional agent which is used to bond chemically the transducer and the recognition element, okay, the transducer surface and the recognition element. So these are some of the methods related to immobilization of the recognition element onto the surface of the biosensor. So the next aspect in sensor design which comes very often is okay, at the end of the day we have to ask the sensor to do a job, okay, and whenever it comes to doing a job, there have to be measures and means to identify whether it is doing its job efficiently. And so for that there are several aspects which one needs to consider for understanding the performance of a particular sensor for trying to take up an analyte. So these are classify under this performance factors. So one of the very important aspects is selectivity, okay, how selectively is this combination of a recognition element, a transducer, and a signal processor is able to pick up a particular analyte of interest. So I would try to define this by saying that selectivity is the ability to distinguish between different substrates. In biology or in chemistry, this word substrate really is a little used in a little different sense. If you talk about microfabrication, substrate is actually the base plate on which you are doing the fabrication, okay. But in chemistry or in biology, substrate is really the molecule which you are trying to target or which you are somehow trying to pick up. In case of a sensor, substrate is that chemical or substrate is that molecule which you want to detect really, okay. So the molecule of interest is also known as the substrate in this case. So the selectivity again is the ability to distinguish between different substrates. So if I have, let's say a pH electrode, so can I distinguish between the hydrogen ions in a medium or the sodium ions and the calcium ions? So this is called the selectivity of the particular electrode. The pH electrode would be designed in a manner that it can always distinguish hydrogen ions from the other available ionic sources which are there. The criteria could be many, it could be on the basis of sizes of those ions, as a matter of fact. There may be a membrane which can pick up only the size of a hydrogen plus ion, which is probably very small, and cannot pick up other moieties like calcium plus 2, which is probably much larger in size. So that is the selectivity aspect of the sensor. Another interesting aspect which comes into consideration is the sensitivity range, and essentially as the name kind of indicates, sensitivity is the ability of a particular sensor to try to find out what is the minimum detection limit or minimum kind of concentration of the analyte that it can sense. So usually sub-millimolar range, but in some cases it can go up to femtomolar range.